All right, well, welcome to Bio 303, a survey of genetics. My name is Praveen Babu. I go by Dr. Babu. And what we're going to do today is start off with a little bit of perspective on genetics. So what we're going to do is go over what to expect for the course. And of course, in terms of topics, I'll introduce you to some of the topics we'll be covering over the entire course. This can range anything from what you may think of as classical genetics, meaning, Gregor Mendel's work, to modern genetics, modern forms of genetics like recombinant DNA technology, where we use genetic engineering to do all sorts of different things with many, many different applications in both medicine and everyday life in general, actually, not just medicine, but in say, for example, bioengineering for bioremediation for cleaning up the environment or something of that nature. So let's get started. One of the main reasons why I really enjoy teaching this class is because genetics is absolutely everywhere. And so you can see that up here. So let me tell you a little bit about how I lecture. So right now we have a bullet point here. You'll see obviously a lot of bullet points, but you'll notice at certain times, certain terms are in color, right? And so those are not just vocabulary terms. I'm not going to just give you a vocabulary test, but those are important terms that are going to be necessary in order to understand the questions that I'll be asking you on quizzes, on exams, etc. And so you want to become familiar with genetic terminology. In addition to that, if you ever see an entire bullet point in color, that'll tell you that you've got something that is really important. And of course, I'll say that. I'll say, oh, this is really important. I'm going to ask you about this. And I'll give you example questions. My goal is to try and make this as straightforward as possible. I really enjoy teaching this class because, obviously the title of the slide, genetics is everywhere, but also because it's interesting not just as a subject of study, but also in that it affects us, in our daily lives on a daily basis, in medicine, in the food that we eat, in all sorts of aspects. So, as the bullet point says, genes influence our lives in many, many ways. And so they are responsible for many things. They're responsible for our height, our weight, our hair color, pigmentation, susceptibility to diseases, disorders. And what's more important here is that you realize that not everything is genetic. So it's not just the things that you can see, like your height or your weight, etc. It's also whether you're lactose intolerant or all sorts of other things, all sorts of other attributes about us. In addition to that, there are a tremendous number of aspects about us that are not 100% genetic. So that's the interesting thing. The very first slide, I'm telling you, genetics is everywhere, but it's not all genetic. So the parts of, like, let's take intelligence, for example. So you are born with genes that allow you to basically have the ability to learn. Right? So these genes contribute to our intelligence and our personality and whatever it may be, but they do not determine how intelligent you will be, right? Obviously, you're in school because you have access to education. This will increase your knowledge, and from that, you can learn and apply that knowledge, and you can increase your intelligence. If you don't have access to that education, well, then, that doesn't mean that you're not going to be intelligent. It just means that you won't have the same tools. You certainly still could be intelligent. And then, of course, you could have all the genes to be intelligent. And unfortunately, things aren't working out for you. And the nice way of saying it is that you just do not utilize those tools. You do not take advantage of those genes, and so you turn out not to be intelligent, right? So, in terms of our personality, for example, all right, so what we have here is an example of a woman who is albino. What that is one of the very, it's a wonderful example of a genetic condition. What that is there's a particular gene. There are actually multiple genes, but there's one particular gene that is responsible for the production, an enzyme involved in the production of melanin. Melanin is what pigments our skin, our eyes, our hair, etc. And if you have a mutation to this one gene, then you cannot produce melanin. So it's a yes or no answer whether you can produce melanin or not. You'll see that many things in human beings and in many organisms are not just going to be like genetic yes or no, but there's also going to be an environmental factor. But in this case, we have something that is purely genetic. This is metabolism. This is basically not being able to produce melanin. And that is a genetic aspect that gives us what we call a phenotype, the overall outcome. In this case, the phenotype would be, you could see she has red eyes. That's because she doesn't have any pigmentation in her iris, and so therefore you could see straight through to the blood vessels in her eyes, and so her eyes appear red. Obviously, she doesn't have very much pigmentation in her hair or skin, etc. And so she, because she doesn't have this melanin, needs to avoid the sun. And what's interesting is that this can also then now affect her personality, because there are a tremendous number of people that are albino, and then because they have to avoid the sun, they end up taking on a lifestyle that is completely the antithesis of the sun. So dark clothing, a dark lifestyle, if you will. And so that would be where you could see how genetics is going to then interact with the environment and therefore be influenced and influence, I should say, our personality. So one thing I was just saying a moment ago is to keep in mind that while genes, you hear this all the time. Oh, yep, it's genetic, right? I have, so I'll be telling you a number of different stories from my personal life, just because I absolutely love teaching this class. My life is one big science experiment, and it's all right. You can laugh at me. I have self-esteem. But basically, what you need to do in the very first slide of this course is, well, I guess second, is to remember that not everything is genetic. All right. So, like for example, let's talk about albinism again. Albinism, if you think about it, should not be prevalent because it does not confer any advantages, right? So here is an old photograph of a young girl, a Native American girl, who has albinism. And so if you go back, we're talking centuries ago, she was a member of basically a hunter-gatherer society, and so working out in the field. So actually, I shouldn't say hunter-gatherer, right? So they domesticated crops and whatnot, but farming, right? So working out in the field, not something that would be good for her, because in the sun she's going to get sunburn, skin cancer, etc. And so one would think that the population would be very limited in terms of albinism, but it's not all genetic, because actually in what is it, the Hopi Nation, I believe, as well as another indigenous people of the Amazon, two very isolated groups, they have a tremendously high rate of albinism. And that's because there's also the aspect of humans, of society. And in their societies, they viewed a child that was born with albinism as an exalted one, a clean individual. And so this child, be it male or female, would not have to toil in the fields or anything like that. They would be treated like royalty. They would be fed. They would be brought, food would be brought to them during the day. The men would leave the village in search of food, and the women would stay behind. And then if you had a male albino, well, then he would have access to all of these women. He would have a much, the parentally, or I guess the delicate way of saying this is that he would have an enhanced genetic fitness. He would have quite a few more offspring, right? And if anybody were to give birth to an albino child, well, then that would be another blessing. And so in those populations, there's a tremendous rate of albinism, even though there's no genetic selection for this albinism. And that's because, again, it's not all genetic. I can give you another example. And I promise I'll go faster on the slides than this. But a long time ago, in our first house, we were remodeling and we were doing a lot of work. And I was up on a scaffold. I was like 25 feet high, and I almost fell off. And so then I thought, well, this is really stupid. I probably should pay somebody to do this, because they have workers' comp. If they fall down, if I fall down, that's it. So I got help, and of course, I still got up there and still kept working because I'm an idiot. And I never fell down. But the guy I was working with said all sorts of horrible, racist, stuff, because we didn't have much to talk about, but we were talking about sports, right? Basically, like we were working side by side in close quarters for days, and so we basically decided we were going to talk about sports, and so we're talking about sports, and he made all sorts of generalizations that were absolutely untrue and just plain racist, and I needed him to do a good job, so I basically was just like, whatever. Since this is a recorded lecture and you don't see me, I have a very dark complexion. I'm of Indian descent. And so, it was a little troublesome to me, but I needed him to do good. I needed him to do a good job, so I tried to keep it in. I didn't say much, But the thing that like just killed me was that eventually I didn't want to tell him what I did for a living. And then eventually he finally got it out of me and he's like, "Oh, you teach genetics. Oh, well then you know that like certain people are just genetically faster or stronger or blah 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 and all sorts of stuff." And I was just like, "No, no, that's not how it works." He's like, "Yeah, it is. You know it is, right?" And so, but then what was interesting is that and everything he said was like, it's all genetics, it's all genetics. Lots of people have that misconception. And so my goal with this course is to try and give you enough information, enough background, so that you can make an educated decision on and have an educated view of what actually is genetic and what's not genetic. And I guess since I went down this road, I might as well tell you the end of the story. So I finally couldn't take it anymore, and the job was almost done. So I just let into him, and I was, the first question I asked him was like, "You know what? This is just horrible. Like, do you even know what a gene is?" And no, he had no idea. People today, still today, do not have a very good idea of what a gene is, but yet they're very comfortable saying that everything is genetic. This is genetic, that's genetic. And I'm here to tell you that, in fact, quite a bit is not genetic. So let's, well, I guess I should have used this statistic here. So OCA2 albinism occurs in 1 in 20,000 individuals normally. However, in certain populations, because of societal pressures, it could be much more prevalent. So you can see it occurs in 1 in 200 individuals among the Hopi Nation, right? So the Hopi Native Americans in Black Mesa, Arizona, as well as about a 1 in 200 individuals, as well as in another indigenous population in a small tribe in the Amazon. And so the reason why, right, and we've already kind of touched on this, the reason why is because, well, here it is in red, right? It's not all genetic, right? It's not just the science, it's also the human nature that has to play a role on that science. So your evolutionary textbook would tell you, well, there's no selection pressure for albinism in human beings. I mean, basically, you would select against it because it causes skin cancer, premature death, blindness, whatever it may be. But in fact, it actually increased in the population among these individuals simply because of their societal pressure. So how our genes interact with the environment is a very interesting twist to genetics. And you'll see that as we go through the course. And so there it is, because we live in societies, societal pressure. You heard me say that a few times. Okay, so remember my question, what is a gene, right? And usually at this point, I poll the audience, what is a gene? We get a number of different answers, right? And that's the point. There are a number of different answers. You could answer generally. You could say it's a unit of heredity. It's something that you pass down. You could be more specific. So that would be an example of like transmission genetics. We'll see in a moment there are different areas of genetics. Transmission genetics is how you study what is being passed on. And if you've ever heard the phrase, oh, that skips a generation, that sort of thing, that would be transmission genetics. So if you say a unit of heredity, you're giving a transmission genetics answer, which is correct, but you could be equally correct and you can give a molecular genetic answer where you're talking about the DNA molecule itself and the biochemistry of it. And you could say, well, a gene is going to be a sequence of you have a polynucleotide, a double helical polynucleotide strand, and then it is composed of multiple nucleotides. We symbolize them based off of which nitrogenous base they have, A, C, G, and T. And so it could be a stretch of sequence that codes for a particular protein or something like that. That would also be another correct answer. There are many correct answers. The more we learn about genetics, the more correct answers there are. But for now, let's just say it is a unit of heredity. So a gene is a unit of heredity, and as I mentioned a moment ago, it certainly can code for something. It can very well not code for something. Either way, it is still information in DNA. So coded information in DNA is pretty straightforward. That's, oh, well, this codes for this particular protein, this enzyme that is necessary for this, metabolic pathway, etc. Whereas non-coding information could be, well, you could also have a regulatory gene. You could have a gene that helps to actually determine when these other genes are going to be turned on or utilized or turned off. So when you utilize a gene, we call that gene expression. And some fundamental concepts. Let me throw some fundamental concepts out there. You've already got one. One is that don't think everything's genetic. The first thing you want to take from your genetics class is that it's not all genetic, right? And number two is I just started to define gene expression. A lot of what I'm talking about will also be defined in bullet points on subsequent slides. I tend to foreshadow in my lectures. I talk about things and then they pop up on the slide. So don't frantically try and write everything down unless, obviously, if there's something that I say I'm going to talk about and you don't see it for a little while, feel free. And then when we do get to it, it'll be, fresher in your mind. But the second fundamental thing that I want you to take from this very, very first lecture is that, number one, it's not all genetic, and number two, when a gene is turned on, when it's utilized, whatever it may be for, in coding for a particular protein or, basically regulating another protein, that's called gene expression. And so when a gene is turned off, it is off. It is not being utilized. Off is off. However, on is one of a multitude of different settings. So the way I tell my students to think about it is that gene expression in general, you should be thinking of like genes being on dimmer switches, right? So like the little rotary knob, old-fashioned dimmer switch where you turn it all the way counterclockwise until it clicks and it's off. And then as soon as you click it and start turning clockwise, it's on. Well, then the lights come up a little, you turn a little more, they come up a little, you turn it all the way. If you crank it all the way to as far as it goes, that's called constitutive gene expression. And so look for that term in the upcoming slides, constitutive, constitutive, constitutive. And so basically what you want to take from that is, number one, it's not all genetic, and number two, genes are on dimmer switches. So when I say on, when I say expressed, don't just think of like a flip switch where it's like on, off, right? Think of a dimmer switch. On doesn't tell you the whole story. On tells you, yeah, it's being utilized, but how much is it being utilized? All right, so now here's another question for you. So normally I would, of course, interact with my students. Obviously, these are strange times in which this is being recorded under the shelter-at-home order for the COVID-19 pandemic. But basically, question is genetics a new field of study? So take a moment, actually try and answer that question. Is it a new field of study? Usually, my large lecture hall classroom, it's pretty split, 50-50, yes and no. And what's fun for me is that it's essentially everybody's correct. Yes, no, both of those are correct answers. So yes and no. Yes, because what you know about now in terms of genetics is relatively new if you think about the history of humankind. And no, because people have been working with the concept of heredity from the beginning of humankind. Let's put this in perspective. So a little bit of background on this first slide, I apologize, but basically, here's your perspective. For the history of humankind, so even before we had written history, people understood that if you had a child with somebody, that child would have traits, would have characteristics that were somewhat like you and somewhat like the other person with which you had that child. And so you were passing on those traits. So the concept of passing on traits, the concept of heredity, has been with humankind from the beginning. Therefore, the idea of trying to explain this has been with humankind since the beginning as well. So if you think of genetics as the study of heredity, that's been around forever. Even if you were to be specific and say, okay, well, no, let's make it scientific. Genetics is, like breeding or, domesticating livestock or crops and then breeding them, etc., and then breeding them to give you certain things. Okay, well, that's still been with us for centuries because obviously we switched from hunter-gatherer to an agrarian society long, long ago. And then if you want to say, is it a new field of study, and you say yes, well, then you would be thinking more of like molecular genetics. We only have had this structure of DNA since 1953, which is not that long ago. If you think of the time scale we're talking about, the entire history of humankind, we've been trying to explain how we pass on our traits, and then in 1953, 1953, we get the structure of the molecule that is the chemical component of heredity, the actual thing that we use to pass on our traits with. And so since 1953, there's been an explosion of molecular genetics, things happening at the biochemical, the molecular level. So yes and no are both correct answers. It just depends on which type of genetics you're talking about. So, to put that in perspective, people have understood the hereditary nature of traits and practiced genetics for thousands of years. And so, like, for example, it's impossible to avoid genetically modified foods. Here's a fun topic that everybody seems to be talking about just ever since I've ever, I can, as long as I can recall. So when I say it's impossible to avoid genetically modified foods, let's think about that for a second. So there's a difference between genetically engineered foods and genetically modified foods. However, the general public does not know the difference. And so we just call them genetically modified foods. And so it is impossible. So all those labels that you see that say non-GMO are mislabeled. They're basically just not. If you look on Polylearn, you will see that I have posted some articles about propositions to label everything that contains genetically modified, ingredients, and that failed. That never even, basically it didn't come close to passing, because once people actually started learning what they would have to label, which is absolutely everything, then they started, taking exemptions for dairy products, taking exemptions for vegetable products, and basically by the time it was all done, there was nothing left, because it was going to cost a tremendous amount of money to slap a label on absolutely everything we consume. It is impossible to avoid genetically modified foods. If you think that when you open up an ear of corn and all of those kernels are big and they're uniform in color and they're juicy and they are perfectly lined up on that cob, and you think that that is in any way natural, you are seriously deluded, because that is not what corn used to look like. That is what corn looks like now. So the most genetically modified crops that I can think of by far are corn, wheat, soy, rice, all of these staples that we have. If you've ever eaten a strawberry in your life, you ate a cloned strawberry, because it is so much easier to just clone the plant and make more plants and plant them next year than to try and, actually reproduce these plants. Cloning, I guess, is a form of reproduction, if you will. But these are not genetically engineered. There's a difference. For example, here we have, I just tried to find something off the, when I'm in the grocery store, it's weird. I take pictures of, like, it just, I don't know. I mean, I guess everybody takes pictures of everything now, so it doesn't matter. People probably think I'm posting stuff, but actually I'm doing scientific research when I'm at the grocery store. And so whenever I see something, I'm just like, oh, I could use that. And not to eat, just in my class. And so here we go. So, like, for example, here's Orville Redenbacher's Smart Pop, right? And so this is microwave popcorn. If you look closely, you can see that it's made with 100% whole grain. First off, 100% whole grain, non-GMO corn. Non-GMO corn is, that's like saying, like, dry water, right? I mean, it doesn't work. That's not possible. And so you can see, and to prove me right, there's a little asterisk there, right? I wonder what that means. And so here's the thing. Genetically engineered is when we go in and we change something. Like, for example, I can totally understand, and this is an actual issue that has come up for many people. If you were to take a porcine gene, which is basically a gene from a pig, and put that into a plant that enhances the growth capabilities of the plant, etc., does that make that plant now a pork product? Right? Does it make it, for example, for Muslims, for Jewish people, is it kosher, right? And so you get the idea that that's an interesting conundrum, but that would be genetically engineered. And so what you could see here is that this is made with 100% whole grain non-GMO corn, asterisk. Let's find out what that asterisk says. Well, there's the asterisk. Let's look very, very, here we go. So way down here, you can see it. Made with non-genetically engineered popcorn, meaning it's not genetically engineered. So you can take that to mean, because it does, that, oh yeah, it's actually GMO. But nobody knows what GMO actually means. So we're just going to say it's non-GMO, and technically we're correct because it's not genetically modified. It is just not made with, it's made with non-genetically, let me try that again. I'm sorry. It is genetically modified. It's just not genetically engineered. What do I mean by genetically modified? Let me make it very simple for you. Imagine you grow corn, right? And let's go way back to when we first switched from hunter-gatherer to agrarian societies, and you're growing corn and you're new at it, and let's say it's a bad year, it's a drought year, and some of your plants grew really well, so you have some corn, others died off. Well, you know what you're going to do? You, if you're smart, are going to take the plants that did well, that were drought tolerant, and you are going to grow them next year, right? You're going to basically choose to grow those next year. And so if you choose to grow those next year, well, then you know what that means? It means that you just genetically modified your stock, right? You introduced what's called artificial selection. That's genetic modification. What we do nowadays in the laboratory in a weekend obviously took an entire growing season, 100 years ago, 1,000 years ago, but it's still genetic modification. You are genetically modifying your crop by decreasing the genetic diversity in your crop by growing one plant or one strain of plant, right? And you chose that plant artificially. It wasn't naturally selected for. You chose it. You sped up the process. That is genetic modification. So, just in case you couldn't read it, here it is: made with non-genetically engineered popcorn. All right. Now, one thing I want to mention is that we are Bio 303. So we're going to talk about the science, we're going to talk about the genetics, we're going to talk basically the biology of it, right? I don't want to dismiss the ethics and morality of a lot about what we're talking about. I have absolutely no problem whatsoever if you choose, after learning what genetically modified is, what genetic engineering is, et cetera, if you choose to disagree with that, and it doesn't, your beliefs are in conflict with that, and so you choose to try and avoid genetically modified foods for that reason, I can respect that 100%. I have no problem with that. But my problem is if you choose to avoid these things simply because somebody told you it was bad, right? And you don't know why. Or, you choose to aim for these things because somebody told you that was good, but you don't know why. That's my problem. So I don't want you to be, well, I would like for you, you're in college, you're learning how to think critically, you're learning how to make your own decisions. That's the point. So take the information that I give you. Obviously, you want to be critical of your source. I'm going to try my best to be objective and try and give you both sides of it, but let's be honest, I love this stuff. And, obviously, no matter how hard I try, I'm going to show a bias towards genetically engineered foods and whatnot because I see it as an improvement. But I'll try and do my best to give you an objective, as objective as I am possible of, account of all of this so you can make your own decisions. Now, with all that being said, keep in mind that genetics, their application of all the technologies we'll be talking about, all of these things give rise to many issues of privacy, ethics, and legality. And the thing is that genetics isn't just science. Genetics is science plus the ethics, whether we should do this, right? It's not a matter of whether we can or cannot. It's a matter of whether we should, because you'd be surprised what we are capable of doing. And then, of course, the legality of it as well. Let's take the cloning of human beings, for an example. That is immoral. That is, any self-respecting scientist can agree that that is something that you should not do. And in fact, it is illegal at this point to try and clone human beings because, let's be honest, you're not perfect. You're not going to get it right on the first try. And every time you make a mistake, that's murder. right? And so I would hope we could all agree that murder is wrong, and so therefore trying to clone an individual, the fact that you're going to make mistakes, that is also going to be wrong because it is inherent in that you will be committing acts of murder. So, genetics and the applications of its technologies give rise to all sorts of issues. Scientists, as well as physicians, as well as politicians, and all sorts of other individuals constantly debate the fate of genetic information and how it is used legally and illegally. And this all stems from the dark time in genetics, and that was its horrific misuse. And that horrific misuse was during the time of eugenics. So let me just give you a quick refresher on what eugenics is, because in my past classes, I've found a very large number of students had no idea what eugenics is. But let me start with this. Let me start with some basic, like observations we can make. If I were to ask you, generally speaking, when there's a movie plot that has something to do with genetics, is it usually in favor of genetics or going to utilize that genetic aspect of the movie plot for something to go horribly wrong, and eventually, usually, something is going to start killing people and eating them or something of that nature, right? That's usually, the latter is usually the case. Genetics is not well received. In fact, that's why, microwave popcorn even advertises that it does not have any genetically modified organisms or whatever, GMO corn, non-GMO corn, even though it does. And so the reason why is because people do not trust genetics. The most common phrase you hear is like, that's playing God, right? And now you might find this very strange to hear from your genetics professor, but there is a good reason for that mistrust of genetics and geneticists. And so that comes from eugenics. So earlier, well, I guess I shouldn't say earlier. So last century, right? So the 20th century, early 20th century, so 1900s. Early 1900s, what we have is this eugenics movement in the United States, and most people don't even know about this. This is a very bad time in American history. So genetics was relatively new, right? So we have Mendel's work being published in the late 1800s, and then being rediscovered in the early 1900s, and then you have this explosion of genetics, this new field of genetics. And you can look on Polylearn. I have posted a documentary about eugenics. It's really short, but it's amazing, and it's very interesting about the history of genetics and eugenics here in the United States. Let me just try and give you a short synopsis of it. So if you're not familiar with what eugenics is, it is when early on, so let's think about after World War I, there were a lot of people that basically had their lives destroyed in Europe, and they started coming over here to the United States. But they weren't bringing a lot of wealth with them because they lost everything after World War I, and from many different countries. But they were coming here, and there were wealthy people here in the United States that wanted to maintain their class structure, wanted to maintain their way of life. And so genetics was also a new science at that time. It was a black box science, but the thing that was really troubling about it was that Mendel's work was gaining traction. And we'll talk about Johann Gregor Mendel and his work in transmission genetics. But the thing that he found was that in pea plants, one gene was responsible for how tall the plant would grow, or one gene was responsible for the color of the peas, right? And so they automatically and erroneously, so here's where it's pseudoscience. So non-scientists were able, and even some scientists, unfortunately, just automatically applied that logic to human beings. And so here in the United States, the goal was to become the most genetically fit you could be. And the goal was, now that we understand genetics, to breed out bad qualities. And so there were, state fairs were a major draw because it's not like they had the internet. And so people would go to state fairs and they would compete for the most genetically fit family. Their genetic fitness would be examined. And to be genetically fit, you couldn't have, and I'm serious, I'm not making these up, you could not have any trace of imbecility or alcoholism or epilepsy, or there was a term called a sex pervert. There was just all sorts of very strange things that they thought you could attribute one gene to. Like, for example, the idea at the time, to give you an idea, that at the time, the idea was if you had, like, if your grandfather built a boat, he was a good boat builder, and he died before you were born, you've never met him or anything like that, and you live in a landlocked state, nowhere near a lake or anything like that, you don't know how to swim, you've never built a boat in your life. The theory was, well, you'd be a good boat builder if you tried because your grandfather was, and there's a gene for a good boat builder, and you have that gene, right? So that was the concept of genetics at the time. And so they used this to try and breed better people. And eugenics was the movement to make sure that we were a far superior race by this measure. Now, here's the thing. So if you were deemed an imbecile or something of that nature, then you could be forcibly sterilized against your will. And these laws are still on the books today in certain states. There are people still alive today, and I'm not talking about 90-year-old people. There are people in their 50s that are still alive today that were sterilized against their will because of eugenics laws. So these laws were really bad. They were basically just discrimination and prejudice guised as science. I should say disguised as science, and they were used to forcibly sterilize many people that were coming into this country as immigrants because they were poor or they were deemed imbeciles, even though there's a classic case. There's two very, very important cases, and I've gone on about this a little longer than I'd meant to, so I'll just try and keep it short. There was one gentleman who was a brilliant professor. He spoke multiple languages. He was fluent in, I think, 4 different languages. He came over from Europe, and he had no money or anything like that. And so they wanted to deem him genetically unfit. So they administered an intelligence exam to him, which he passed really, with flying colors because it was in a language that he spoke and was literate. Then they said, oh, well, no, sorry, that can't be right. They tried again with a different test, different language. He still spoke and was fluent in that language and literate, and so he did well again. He did well again, and then finally found some language, I can't remember what, that he, could take this test in, and he failed it because he couldn't read or write that language, and so they deemed him genetically unfit. That's one example. Carrie Buck is the most, she was in the, extensively discussed in the documentary. She's the poster child for eugenics because this poor woman was in foster homes. She basically had an illegitimate pregnancy. She gave birth to a daughter. She was deemed a sex pervert for that. Her daughter was, and an imbecile. Her daughter was deemed an imbecile, even though later on she ended up going to school and doing quite well. There was nothing wrong with her daughter mentally. And of course, in fact, it later became known that she was not in any way a sexual deviant or anything like that. She was raped by one of her foster parents. And so, that's why she had an illegitimate pregnancy. And as you know, back then, an illegitimate pregnancy, a pregnancy outside of wedlock was, extremely taboo. So it's a really dark time in genetics. Because it was used for all sorts of heinous acts. In fact, it was so bad that Nazi Germany, well, the Nazi movement before they took over Germany, they started praising these eugenics laws in the United States. The University of Heidelberg was giving like honorary degrees to eugenic scientists here in the United States. It wasn't until the atrocities of World War II and the extreme to which they took eugenics and, forget eugenics, just the extermination of people that they did not deem fit to survive or to I don't know, basically just exterminate people because of prejudice, right? And discrimination. It wasn't until that eugenics lost favor, because if the Nazis were doing it, it was so bad that finally people in the United States realized, wait, they got that idea from us. That's not good. And so it lost favor. So I spent a lot of time on this slide. I apologize, but please do take a look at the documentary. The reason why there is so much mistrust with genetics is because of, you may not be aware of this, but it's because of that eugenics movement here in the United States. And so I totally understand when people, have, misgivings about genetics, or when I tell them that I'm a genetic engineer, etc. They, give me the side eye. Some people find it fascinating, and then others do not, right? They think that I'm basically the Antichrist. And I have no problem with that because I'm not. And also because, I actually understand where that mistrust is coming from. So, that all being said, that is the one slide. This slide is the one slide we're going to be talking about the ethics and morality of it. I'm not trying to gloss over it. We simply don't have the time to cover it. You could do an entire course about the morality, the ethics of all of the different aspects of what we'll be talking about in terms of transmission genetics, manipulating that transmission genetics, molecular genetics, manipulating molecular genetics, not just to, help us and benefit humankind by producing, like, for example, producing a subunit vaccine, taking a small portion of a virulent, bacterium or virus, and just expressing a very small portion of its outer viral coat, the protein coat, so that you can inject that into individuals, and then they will develop antibodies against that, so that when they get exposed to the real thing, they will have antibodies already against it. That would be, obviously something commendable, but I can understand why there's still, ethics issues to discuss about that. So addressing those legal, ethical, and social questions associated with all of the aspects of genetics that we'll be talking about is just as important. I cannot stress this enough. It is just as important as the information gained from it. We simply don't, we're a BIO 303, so we don't have the time to actually go over those things. So do not get me wrong. Do not think that I'm saying, hey, all of this is good, all of this is great. I understand why it needs to be discussed and determined whether certain things should or should not be done. In fact, scientists got together early on when they realized that you can cut and paste DNA and start mixing and matching DNA from different organisms, and set down some ground rules at what was called the Asilomar Conference. So the wrong ideas about genetics have influenced past laws that are still on the books today, unfortunately, past court decisions, right? So the Carrie Buck situation, Supreme Court Justice Oliver Wendell Holmes has a famous quote, at least in the genetics realms, about how 2 generations of imbeciles is enough, and upheld the idea that eugenics should forcibly sterilize Carrie Buck and her daughter so that they couldn't reproduce any further, right? So eugenics has influenced past laws, past court decisions. They are still propagated today. If you were to click the link, you would see that it is for, it takes you to a page discussing a movie about Darwin, Charles Darwin. And it was deemed too controversial. The article is titled, that the movie was deemed, I don't remember the exact title, but it was deemed too controversial for release in the United States because it dealt with evolution. And we're talking about, just about, I think it was in the 2010s that this was going on, and so just this past decade, and then once it was deemed too controversial, of course, people wanted to see it, so it came over here. I was pretty excited I went to see it, and it turned out to be a horrible movie. It was all about, it was a drama. There was no science. It was basically it was not what I expected. I have nothing against dramas, but it was basically about whether Charles Darwin should or should not proceed with his Origin of Species, and how he was fiercely religious, and what was his wife going to think, and it was very slow. It was a very slow movie. But my point is the fact that movie, just being about Charles Darwin, it wasn't really much about evolution. Trust me, I saw it. But why would that be controversial? It's obviously, just saying evolution is still controversial here in the United States. Even though evolution is not a theory anymore, it has been proven, it's been demonstrated, and you can look at a time-lapse video and watch evolution occur with bacteria, because they rapidly, they turn over so rapidly. It's harder to see with organisms that have a longer lifespan, but you can certainly see that. But unfortunately, it's still controversial today. So my goal in this course, right? So I apologize. This is a very long slide. My goal in this course will help you make informed decisions regarding the many genetic issues that will arise in your lifetime. And so take everything I say with a grain of salt. I believe in peer review. I believe in critical thinking. And so you can certainly say, well, he's pretty biased and he really likes this stuff. But what I'm going to do is present to you facts, and you can interpret them however you wish. So let's get started then. So my question up top here is, what is genetics? Well, we've already heard genetics is the scientific study of heredity. So it's a scientific study. So we want to be quantitative. We want to actually determine how we pass on our traits, not philosophize, not theorize, but actually quantify heredity itself. And so genetics determines how traits, and therefore, not just traits. So traits are things like your height, your eye color, your blood type, but traits and diseases are passed from generation to generation. So if we know how, like, say, for example, a particular mutation can lead to a lack of a particular enzyme that can lead to a particular disease, then we might be able to rectify that. If we know how it's passed on, we can gauge the likelihood of offspring having it, and then we can also attempt to rectify that. So genetics examines the molecules that comprise genes, so the DNA, right? And how genes are regulated, so how they're turned on and off. And you might recall I mentioned genes. Think of them as on dimmer switches. Gene expression is highly regulated. Think of your genes. The human genome has somewhere between 20 and 25,000 genes. We're not sure exactly how many genes, simply because they haven't all been checked, right? We have candidates for genes. But let's say we have 25,000 genes. So that would be like an orchestra with 25,000 different instruments, etc. And then it has to be played such that you hear a unified music, right? And that means that some of the instruments are going to be played at different times, some are going to be played at different intensities, et cetera, et cetera. That would be gene expression, and highly regulating that gene expression, in which all those dimmer switches have to be turned a certain amount at a certain time in a certain cell type in a certain part of the body. That's all highly regulated. And so the genes not only encode things, but they also interact with the environment and regulate themselves. So therefore, there are many specialized fields within genetics. And so let's start with population genetics. So let's go with a macroscopic view, and then we'll go down to the microscopic view. So just to give you an idea of what's coming, population genetics, then let's see, transmission genetics, then cytogenetics, and then molecular genetics. But let's start with population genetics, because we're not going to have a specific chapter in this course devoted towards population genetics. It's just going to be peppered in here and there. So population genetics is a field of genetics in which it's a lot of statistics applied to genetics. We gather data, right? Like, for example, during the SARS-CoV-2 pandemic that is going on currently as we're recording this, you might be wondering why. Why are people getting tested to see if they have the disease? And so some people just want to know, but the reason why epidemiologists want those tests done is so that they can keep track of the increasing number of new cases, the decreasing number of cases, et cetera, so they can track not only its spread, but they can gather statistical data. That statistical data is useful because it can later then be applied to, epidemiology. But in the case of other genetic diseases, we could use population genetics in that respect. I'll give you a perfect example. So you're going to hear a lot of examples from my personal life, and so I apologize for that, but it's just how I relate to my field. So my wife and I are very different. So I am of Indian descent. She is of Irish descent. So not only do we look very different, but we are, basically genetically quite different in terms of the alleles of genes, the flavors of genes that we carry. All human beings are genetically the same. There is only one race of human beings on the planet. There's no such thing as race genetically. We're all human beings, Homo sapiens. And so you could take a healthy human, male or female, from anywhere on the planet, healthy human, I guess you could take a healthy human female from anywhere on the planet, healthy human male from anywhere on the planet. They can have a child together because they're genetically compatible. They're part of the same race. Race, as you know it, is basically so it's cultural, right? And so you have these different ethnicities. You might be thinking, well, no, that's not true, because some people are very fair skinned, some people are very dark skinned. But again, remember, whether you're fair skinned or dark skinned, unless you're albino, you're making melanin. It's just one person's making more melanin than the other person. And so it's not really that different. We're not that different from one another. But the differences that you do see are due to the flavors of genes that we carry. We all carry the same genes. That's why when they sequenced the human genome, they didn't have to sequence one person from every different part of the world. They only sequenced a few people. We all carry the same genes, but we carry different flavors of those genes, right? And so when my wife and I were having our twin daughters, so I have this little science experiment going on at home. It's pretty awesome. So my wife is a fraternal twin, and my daughters, our children, are fraternal twins. And so before you say, oh, I thought it skipped a generation, that's just like putting a gun to my head. Please don't say that. So it doesn't skip a generation. Maternal twinning happens. It's possible to happen every generation. It's just statistically unlikely. And so I am just extremely lucky that not only, am I lucky that I have my two little girls, but dude, I mean, it's like perfect material for what I teach, right? So, but let me get back to it. So population genetics. So when my wife was pregnant with our twins, when you're pregnant with twins, you're automatically a high-risk pregnancy. It doesn't matter what your age is. If you're 35 or older when you're pregnant, here in the state of California, that's deemed a high-risk pregnancy. But this was a while ago. We were under that. She was under the age of 35, so that wasn't what made us high risk. What makes you high risk is because it's a multiple pregnancy, so it's twinning. So she's a high risk pregnancy, and so there were all these extra tests that we had to do. And so we went to, our doctor told us to go, so we went to the phlebotomist to get blood drawn for all these tests. And so we're sitting there just waiting, and the phlebotomist comes in, and she's rolling in this cart, and there are literally, I think, about 30, maybe 40 vials that need to be filled. So I was just like, my first joke was basically, how are we going to get home? We're both going to be passed out from donating all this blood. And then I said, so which ones are for her? Which ones are for me? Because I had to have my blood drawn too. And she's like, oh no, these are all for her. Those are for you. And so she showed me like 2 that were for me. And so internally, right, I was just like, yes. But externally, I was like, oh, I'm so sorry, right? So anyway, the point here is that we had our blood drawn. We had this test done. Many tests, and she had way more tests than I did, because obviously I just had 2 vials drawn, so they just checked to make sure I was still alive. And so one of the things that came back from our test was that my wife is a carrier for cystic fibrosis. So this may not make sense to you just right yet, but it's certainly hopefully some of it sounds familiar. So she's heterozygous, so she carries 1 good copy of the gene, 1 mutated copy. So she carries 1 copy of we have 2 copies of every one of our genes. We are diploid, and so that means that we carry 2 copies, right? We have pairs of chromosomes, and we carry these 2 copies, and if that doesn't make sense, don't worry, we'll go over it later. But so she has 1 good copy, 1 bad copy. Obviously, she doesn't have cystic fibrosis. We would have known by now. And so she came out positive as a carrier for cystic fibrosis. Now we're having twins, and so there is a possibility that one or both of them could have cystic fibrosis if I also was a carrier. But in my, very, very, very Spartan testing that was done on me, they did not check for cystic fibrosis or any of that stuff. And so I proceeded to calmly tell her, because I'm an expert in this sort of thing, that, there's no way, right? So I'm, like I told you, I'm of Indian descent. I'm born and raised here in the United States, but my parents are from India. And so I said, oh, look, so what you're looking at is actually a map that I took. You can see here's the website from where I got it. But I'm Asian. I'm in the Asian category. And so I told her that, there's like a 1 in 350,000. I even had the numbers memorized back then. So 1 in 350,000 chance that I have the cystic fibrosis allele. And I'd only have 1 allele, because obviously I don't have cystic fibrosis. alleles. And so even if I had one allele, then on top of that, you would need, statistically, you would need my allele and her allele to end up in one of our, or both of our two children. And so like statistically, it's completely unlikely because she is of European descent and she has at the time I said 1 in 3,000 chance. Right. I think I said 1 in 3,000. You're like 1 in 3,000. I'm like 1 in 300,000. It's fine. Don't worry about it. But of course, this is my wife, and she's pregnant with twins, and so and it doesn't matter that I'm an expert in this. She basically says, "Nope, you're getting tested." And so I got tested, and guess what? I am not a carrier. Imagine that. And it's not like I could scream, "I told you so," or anything like that. But I kind of told her so. But I didn't say that to her. And so basically, how would I know these numbers? Right? The point to all of this, if you're still with me, is population genetics. That's how we would know these things. Over time, we gather data and we start to see trends. And so population genetics is not just useful for predicting like, like the likelihood of particular diseases or anything like that. But population genetics also will allow us to track, animals if we're trying to do some sort of conservation. It'll allow us to determine migration patterns and speciation and all sorts of different things. So population genetics is very general, though. If we talk about like macroscopic to microscopic, this is like the view from 30,000 feet. You do not see individual people from 30,000 feet high up in the sky, right? So that's population genetics, and a broad overall view. And you can see I'm showing a map of the entire world to tell you about population genetics. But then the opposite end of the spectrum would be biotechnology. Biotechnology is what you might be thinking of when I say genetics, because that's the newer, cooler stuff, right? So that's going to be when you're dealing with the individual genes. So you can take a gene from absolutely any organism on this planet, because we all speak the same language. We're all using the same operating system. So you could take like a gene from like the human gene for insulin. I'll pick that one because that one has special significance. It was the first one that was subcloned. And so you could take the human gene for insulin, and then you could like cut that out of a human cell, and then you could plug that into a bacterial cell, say like E. coli, the same stuff you get at like Taco Bell or Chipotle. And so you can plug it into E. coli, and if you do that, E. coli can make insulin. Now, E. coli has absolutely no need to make insulin, but you can make the E. coli make insulin for you. It makes so much insulin, it actually solidifies as a little brick. It crystallizes out inside the E. coli cell, and then you can purify that insulin from those E. coli cells, and now you have actually it's called recombinant human insulin, or Humulin is its trade name. And now diabetics have a source of insulin, a cheap source of insulin that is safe to use, because prior to that, so this happened in the late 70s, early 80s. So there was a group, Stanley Cohen's lab, Herbert Boyer's lab, so all these groups up in the Bay Area, right? So you had a group from Stanford, you had a group from UC Berkeley, you had a group from UCSF, and they all ended up meeting at a conference in Hawaii and discussing what they were doing. And one group knew how to cut a gene out, and then the other group knew how to paste the gene back in, and then another group knew how to take a gene and introduce it into a bacterial cell. And so they all got together and they figured out a way to clone the human gene for insulin and put it into E. coli and produce Humulin, which is how Genentech got its start. And they obviously made quite a bit of money from Genentech, but the point is, but prior to that, diabetics had to get their insulin that was just isolated from like the slaughtered pancreas of sheep, cows, and pigs, and that's not human insulin. So it could have pathogens in it if it wasn't clean, but let's say it was. But over time, you're injecting yourself with a foreign insulin, and your body will eventually recognize that it is not human insulin because it is slightly different, and then it will develop antibodies against it, and then you'll be allergic to it. And so let's say you started with like sheep insulin, and then eventually you're allergic to it. And so then you switch to cow insulin, and then, years go by, and then eventually you're allergic to that. And then you switch to pig insulin, and then years go by and you're allergic to that. Well, that's it. You don't have any other options. And so without insulin, obviously your quality of life decreases tremendously, and then eventually that could lead to a premature death. So human insulin, on the other hand, there's no chance of any sort of, well, I shouldn't say no chance, but there's minuscule chance of rejection, and so you can live a much longer, more healthy life because of it. So genetics is a very broad field from 30,000 feet down to the sub-microscopic molecular level. And so genetics is a very broad field because it is the key to all of biology. And you may have heard this many times before, genes are the blueprint of life, but there's actually some truth to that. So here it is, DNA is the blueprint of life. And so DNA, right? So there's the DNA molecule. It's a double helix. It's two polynucleotide strands wound together to form a double helix. DNA stands for deoxyribonucleic acid. And let's be realistic. DNA is just a string of four types of chemical subunits, right? So A, C, G, and T. That's it. It's a long string of it. It might sound weird to you that your genetics professor has told you, number one, yeah, it's not all genetic. That's not that important. And here's another one. DNA is not that big a deal. It is not that amazing of a molecule. It is just what nature has chosen to use it for that makes it so amazing and interesting to us. But it's actually the point of it is that it is a rather simplistic molecule. It is a sturdy molecule. It is repeating. It is not some crazy unique structure. It is just a polymer that's two polynucleotide strands twisted around one another and they hydrogen bond. And you have eaten, for example, metric tons of DNA in your lifetime, and you will eat more. And you just digest it and you break it up and you use it for parts. So if you're ever worried about those people that are trying so desperately to avoid genetically modified foods, why? Right? I mean, like, are they honestly? Many of them are afraid that it's going to affect them in some way. But let's be realistic. You have always been eating the DNA of all sorts of things and they haven't affected you. You don't eat an apple and consume apple DNA and worry about sprouting leaves and roots, right? I mean, technically, everybody's a cannibal. You're eating your own DNA because skin cells slough off on the inside of your mouth. And so, there's that aspect of it. You can do, I guess that's kind of a pointless statement. I apologize. It's information that you could use. I have plenty of useless information too. Don't worry. But basically, it will not be on the test. I will not ask you if you're a cannibal. But basically, the order of the four different subunits, right? The genetic code. That's what's interesting, because if you have four subunits, A, C, G, and T, if you use those and you make a really long molecule, you can encode a tremendous amount of information. In fact, you can encode the entire diversity of life, all the life that you see, plus all the life you don't see, all that microorganism, all the microbiology, all of that stuff, right? And so think about how much life there is on this planet. I mean, there are entire channels devoted towards telling you about how quirky life is. And you think of the Discovery Channel, right? There's always something on the Discovery Channel where you're like, oh, wow, look at that. And so there's tremendous diversity in the life that we see on the planet, but every single living organism utilizes the same genetic code to encode its information. And so that's the beauty of this. We can mix and match. Do you see the potential from a genetic engineering point of view? Obviously, if you're against genetic engineering, this is frightening. But from a genetic engineering point of view, really the only limit is going to be whether we should, not whether we could, because you could find a gene that codes for, I mean, obviously you need to remain within the realm of physics, right? So, you're not going to just I don't know, like take the X-Men, right? You're not going to have some gene mutation that gives you the ability to fly, right? I mean, there's physics involved here that's not going to happen. But within the laws of physics, if we're bounded by the laws of physics, then you could take a gene for, you could find a gene that codes for something somewhere in some organism and put that into another organism, and that organism could very well use that. I mean, it can read the gene just fine, and it could try and attempt to use that information, and you could confer that ability to that organism. So, for example, something as simple as that E. coli cell could be obliterated by a very simple antibiotic like ampicillin. But you could then introduce an ampicillin resistance gene into that organism, and now ampicillin is not something that could kill it off. So, genetics and genes, they dictate what each and every cell is capable of doing. And since the cell in cell theory tells us that the cell is the basic unit of life, in biology there are 5 criteria that need to be met in order to constitute a biological organism. This is not philosophy. This question, what is life, is a very simple question in biology, because in order to study the ology of life, biology, you have to define what life is. So those 5 criteria are: life is organized, life utilizes energy, biology, life grows, reproduces, develop, life evolves, and life maintains internal constancy or homeostasis. Those 5 things cannot be achieved unless you have at least a cell. So a cell is the basic unit of life. And because genetics or genes dictate what every cell is capable of doing, so all the tangible assets that cell can make within it, right, all the enzymes, all the particles, all the molecules, all the organelles, all that stuff, all those tangible assets are encoded by genes. And then on top of that, how those cells interact with the environment are also going to be encoded by many of the genes that it has. That is why we say that DNA is the blueprint of life, or I heard it as DNA is the blueprint of life, because it dictates what every cell can or cannot do. So genetics controls cellular function and therefore links living organisms. So hopefully, so you can see, here's an example of a statement in all blue, right? So it doesn't matter what color, I just pick a random color scheme for the slides. But you can see here's a statement, here's a couple statements, right? In all blue. So in all, whatever color for the color scheme of the slide. And for those of you that may be colorblind, they're also bolded and shadowed. So those are really important points. And so the point that genetics controls cellular function is how it links living organisms, all living organisms, because genes link all living organisms, is an important point. Genetics is something that links all living organisms, and it is the way in which we pass on our traits. It dictates how each and every cell is, or what each and every cell is capable of doing. All right, so genetic principles are therefore universal, and so they are not exclusive to just people. I'm going to use people as an example. I'm going to use human beings as an example many times over, but I recognize we have a tremendous number of different majors in here. We're usually dominated, this class is dominated with the animal science majors, but we have as well, many other people as well, and so I will make sure to discuss not just human genetics, but the genetics of other organisms as well. But I start off using human examples because obviously we can all relate to that. So modern genetics actually is a confluence of all sorts of advances made while studying many different organisms. So I mentioned bacteria already, but then bacteria are not too much like us, right? Bacteria are usually, they're single-celled and they're prokaryotic life, meaning they don't even have a nucleus. We have nuclei. So yeast are actually a really good organism to study because they are eukaryotic. They have a nucleus, but they're single-celled, so we can kind of grow them like bacteria, grow them really quickly, in large numbers in a laboratory setting, et cetera, et cetera. So we look at single-celled prokaryotes, single-celled eukaryotes, and then animals, right? So insects count as animals, plants, all sorts of different things. And by studying all these different organisms, we can learn a lot about those organisms and then apply what we learn to human cells and human beings as well. So we can divide modern genetics into 4 main classes. And now this is important. Let me say it right now. Your book has gone back to 3 classes. So one of these classes is not in your textbook. It used to be in a previous version. Then I took it out. I have no idea why. I will show you why. It is still very relevant and we still utilize it extensively, and so it should still remain as one of its 4 main classes. So let's start with transmission genetics. We've actually mentioned one of them already, population genetics, but transmission genetics, we'll start there. Transmission genetics is it studies the mechanisms by which traits are passed from parents to offspring. So you could study how, Mendel started transmission genetics by studying how traits were passed from pea plant to pea plant to pea plant, generation after generation after generation. But then, of course, if you've ever, done any sort of genealogy work where you're trying to learn about yourself, well, if you put a biological spin on it, a genetic spin on it, it would be called pedigrees. And if you've ever looked at pedigrees, you would see how we pass on our traits, or how, if you're breeding animals, you look at their pedigree to see what traits are being passed on and trying to perhaps breed out certain traits or, maintain certain traits. Then we have cytogenetics. Cytogenetics is the main class of genetics that your book no longer has in the main classes of genetics. It omits cytogenetics now. I don't know why. Cytogenetics is very important. Cytogenetics studies the organization and arrangement of genes and chromosomes via microscopy. So this is so if I said that population genetics was from 30,000 feet, well then cytogenetics would be not at the molecular level where we're looking at individual genes and DNA sequences, but we are looking under a microscope. So sort of at the like cellular level, right? So we're looking at the chromosomes and what they do. And you can learn a lot from looking at a chromosome from far away as opposed to being able to sequence that DNA in that chromosome and learning from the sequence as well. If you were to sequence that DNA, you would be talking about molecular genetics. So that studies genetic events at the biochemical level. So we're talking at the at the molecular level, the DNA molecule level. So A, C, G, and T bases are individual nitrogenous bases that are part of a nucleotide, which is the building block of polynucleotides, et cetera, et cetera. So understanding, we'll go over DNA replication, transcription, translation, I should say DNA replication, transcription, RNA processing, translation, post-translational modification. All those things are at the molecular level. And then, of course, we have population genetics. So population genetics studies the distribution and variation of genes in populations. And so what I could say is that if we were talking about from macroscopic to microscopic, population genetics is very broad, very general. Then you get to transmission genetics because you're going from like, generation to generation. So that's still fairly broad. Then you go to cytogenetics. So now you're looking at one organism at the cellular level. And then you go to the sub-microscopic level. So that would be molecular genetics. So now what I'd like to do is just kind of give you a brief introduction to what these different fields of genetics are. As you can see in the upper right corner, right, they talk. You can see I've annotated what chapters we'll be going over this in more detail. But a moment ago I told you that we use genetics as a confluence of information that we've gained from experimentation with many different organisms. Well, geneticists use experimental organisms to study how traits are passed from parents to offspring, right? So we can't use humans for a number of reasons. So let me back up a second. We don't experimentally use humans for a number of reasons. Number one, let's start with the creepiness factor, right? It would be just forget about the legality and even the morality of having controlled matings and seeing what happens when this person has a child with that person, etc. I mean, that is beyond creepy, and there's a whole lot of other problems with that as well, right? But even if none of that was your concern, right? You're just like this, emotionless robot. Then you still have the problem with the generation time, right? Like you don't get your answer. It's not like don't think 9 months. You don't get your answer at birth, right? What if you're trying to study the graying of hair? Right? You're looking at least 20, 30, 40 years before you get any usable data. And then on top of that, your sample size, your ends are going to be tiny because usually you get one offspring. So human beings are not a good choice for experimental study. Now, we certainly, use genetic history. We interview people, we get medical information, and we build a family history and a pedigree and look backwards. We look retroactively and we try and determine things with human beings. But if we're experimentally trying to determine different aspects of genetics or transmission genetics, then we can use things like fruit flies, right? Drosophila melanogaster. These are fruit flies. What's interesting, I've noticed that in a laboratory setting, I treat them with dignity and respect, but at home, when they're, flying around my bananas, I turn into a mass murderer and try to kill them all. And so I probably shouldn't do that, but it struck me as, it disturbed me how much they just annoy the hell out of me, basically. And so I realized that, I went a little overboard because I built a trap, which you can Google it. That's what the internet's for. And so you put like apple cider vinegar in like a little mason jar, and then you put saran wrap over it, and you poke a few holes so they can get in. They smell the vinegar, right? And they can get in, but they can't get out. And so then you end up like catching them all. And so I can't tell you how much pleasure I got from doing that because the fruit flies were driving me crazy at home. And then I realized that, like, in a laboratory setting, I would never do anything like that because, these are experimental organisms and blah, blah, blah. So you have fruit flies. You have E. coli. We talked about E. coli. You have C. elegans. So C. elegans is a soil nematode, right? It's just a little worm that you can find in soil. But the thing that's neat about it is that it just by chance, it was the first animal that ever had its genome sequenced. But basically, it's so small that if you put it on just a dissecting microscope, you can see these with the naked eye, right? And so like this C. elegans worm would actually be like that. It would be about the size of the eye here without the dot. Or let's go with the S, right? So it would be like the size of this S in real life if you were looking at it without any magnification. But if you do look at it with a dissecting microscope and some light underneath, you can actually see through it. And so you can see different processes occurring in the worm because it's basically translucent or transparent even. And then of course we use, Arabidopsis, we use mice, we use rats, and we use, I already mentioned baker's yeast, right? So Saccharomyces cerevisiae. There's other strains of yeast that we use. So Schizo, Saccharomyces palmbae, all sorts of different types of, yeast cells because they're fungus, but they're also single-celled eukaryotic organisms. We can learn a lot from them too. Let me give you an idea of what we can actually learn from these organisms. So this is, if you are actually looking at the slides, the PowerPoint slides and not this lecture video, then you could see, I don't know if you could see that, but basically this is a linked image. A lot of these images have links, and you can just click the link. And so this is it's an older article. It's from 2006, but it's about a scientist, a maverick scientist, who basically was doing some work, and then he got jealous. So you can see the envy. He got jealous of the people across the hall from him doing genetic work with other organisms. And so he dropped everything, and then you can see right here, a maverick scientist is risking all to study the behavior of fruit flies, convinced that they have much to teach us about the evolution and neurobiology of emotions. So this guy is absolutely brilliant, but he switched fields and started studying emotions in fruit flies. And what's really interesting is that, so let me ask you this question, and actually do try and answer it, right? And obviously there's no penalty, nobody can hear you if you get it wrong. But what emotion do you know fruit flies have? Now here's the interesting thing. Every single time I've asked my course, my students this question, whenever I've taught this course in large lecture halls, every quarter I get hunger as the first response. That might be because this class is normally taught, from 6 to 8 p.m. But every single quarter, I when somebody says that, I say, is hunger really an emotion? I mean, I can understand if you're hangry or something, but hunger is not an emotion, right? We have that little side discussion. And then we get to the answer, and the answer is fear. Fear is an emotion. You know that these fruit flies have preservation of life. I know this because every time I swat at them, they tend to get away. So they're trying to get away. And so basically, he decided he's going to study fear in fruit flies, right? So he's going to study human emotions, the neurobiology of emotions using fruit flies. And you can do that. That is actually a viable course of study. You can learn a lot from other organisms because, again, whatever you learn about it, whatever you learn from these organisms, you can then apply them to human beings. You can test that theory, test for either the same gene or a very similar gene in human beings. So that would be an example of transmission genetics, how you pass on, certain traits, and basically you can study many different organisms to learn how they pass them on, and then see how we pass them on. So now for humans, though, we would have to use pedigree analysis. And so pedigree analysis is obviously a much better way to go than controlled matings of human beings. Pedigree analysis would be to retroactively look at family history. So what we do in pedigree analysis is that we analyze, so pedigree analysis utilizes detailed family histories to reconstruct the pattern followed by a trait over generations. This is very difficult to construct. So let me show you what you're looking at. You are looking at a pedigree. So this is your, I guess these would be like the grandchildren. These are the grandparents. First generation would be on top. We call these the P1 generation. We'll go over pedigrees in much more detail later on. But for right now, first generation, right, the P1 generation. And so males are squares, females are circles. They go in age, right? So like, for example, their offspring, firstborn, secondborn, thirdborn, fourthborn. So youngest, right? So in this case, he is older than her because he was born first, right? If she was older than him, then obviously you'd flip-flop. And so then when you have a trait that you're trying to follow and you find an individual with that trait, you basically shade them in. And so this would be an affected phenotype is what we call that. Phenotype is just the overall outcome. Genotype, these are all terms I'll define for you later, but genotype is the genetics that you have, the genes that you have, but you don't know what the outcome is. Because sometimes you can, like for example, my wife is a carrier of cystic fibrosis, but she doesn't have cystic fibrosis. So if we were doing a pedigree of cystic fibrosis, she would not be shaded in, but because this is the phenotype. So this is the affected phenotype. And then we have affected, non-affected, non-affected. All right, so there's quite a few other symbols and things that we need to talk about, obviously. So this would be a mating, right? And so they had offspring, and here's a mating, and so they have offspring, right? We use, kind of sterile terms. You try not to make it too, like, oh, hey, they had kids, right? But basically, and so this one's not a good pedigree because it's not very well labeled, right? It doesn't say P1. This would be the first filial generation, so the F1s. These would be the F2s, etc. And so what happens is, let's say that you want to know what the likelihood. Let's say that, for example, she is about to have children. And she would like to know what's the chances that her children are going to have the affected phenotype, because it might be something that is debilitating, might be something that concerns her. The biggest thing we can get from pedigree analysis is that we could assess the risk for future generations. And we could see that, for example, and we'll be able to do this a little bit later on, but some of you might have some experience. So let's just have a little fun with the pedigree for a moment. So these two individuals, we would say, are both carriers, right? So they don't show the affected phenotype, but they must be carrying it because they have an offspring that has the affected phenotype. And so the most likely scenario is that it is a recessive phenotype where you have to have 2 copies of it in order for the phenotype to show up. And so here's a carrier that has 1 good copy, 1 bad copy, 1 good copy, 1 bad copy, and then here this individual has 2 bad copies, and so they have the affected phenotype. The daughter, on the other hand, she must be a carrier, so she must have gotten, like, let's say, 1 good copy from mom, 1 bad copy from dad. And so she's got, say, for example, she's a carrier of this particular ailment. It doesn't have to be anything negative. It doesn't have to be a disease. I just chose that because that's usually when people are asking about it. And so, she has children with, she has a lot of children, 1, 2, 3, 4, 5, 6, 7. She's got 7 children with him. And so what you can see is that he's not affected and she is a carrier at best, right? I mean, there are other things we'll learn about later on in the course called penetrance and expressivity. You don't need to know what those are right now, but I'm just, sowing the seeds for that. When we do talk about them later on, you'll be like, oh, I've heard that before. And so let's just say that she's a carrier, but she doesn't have it. He must also be a carrier because look, 1, 2, 3, 4, 5, 5 of their children out of 7 end up with this particular affected phenotype. So the most likely scenario is that he's a carrier as well, right? And then there's other things like, well, does it only affect males, right? And so we don't know for certain, but we do know this. So we'll learn about the Y chromosome and how men can pass the X chromosome on to their daughters and the Y chromosome on to their sons. And so if it was something on his Y chromosome, all 3 of these sons should have the same outcome, but they don't. So we can rule out the Y chromosome. We could do a lot of logic. If you've ever told anybody you're taking a genetics course and usually they're like, oh, that sounds really cool, or oh, that sounds horrible, right? And so the bad rep that genetics gets is that it's so complicated, and it's not. It's just, it requires a lot of logic. And so if you like Sudoku, if you like logic games, then boy, is this the class for you. When we get to pedigree analysis, that's that's going to be all logical deduction. So in a pedigree, like I said, males are squares, females are circles, darker symbols indicate those expressing the trait being studied, and the goal here is to assess risk to future generations. All right, now let's talk about cytogenetics. So cytogenetics is the study of chromosome number and structure using karyotyping. So we'll talk about karyotyping here in just a second. This is not a karyotype just yet. And so it's using karyotyping. And again, cytogenetics is the branch of classical genetics, or sorry, of the four main branches of genetics. It's one of the four, the one that is omitted from your book now. So karyotyping is basically a method to visualize and analyze chromosomes. And so what you're looking at are some chromosomes. So these are chromosomes that have been, they've been condensed down. So if you've learned anything about, say, in Bio 161 about the cell cycle, they've already been replicated during S phase of interphase. And so when you get to prophase, they condense down. Prophase of M phase. And so they're condensed in their most condensed state at this point. And so they are then going to be, so you take an image, the old-fashioned way of doing this would be to take a picture and then cut out each one of these little chromosomes and then start to align them by their banding pattern, right? So they've been stained with, in this case, the old-fashioned stain was Gemsa stain, by their banding, their size, their banding pattern, and then their pinch point, right, where their centromere is located. So you can see there's one here, there's one right there, right? And so then you would be able to align these and you'd be able to basically list out all the chromosomes that you have. So here's what a karyotype used to look like. So this, again, is an old-fashioned karyotype. You can see it's black and white, even though the Gemsa stain actually is actually somewhat of this color. It's a dark blue color. And so the chromosomes are then aligned by their number, right? And so the way they're numbered is in size order. So chromosome 1 is the biggest, chromosome 2 is the second biggest, all the way down to chromosome 22, which is the 22nd biggest, or the smallest of the chromosomes. And so the numbered chromosomes shown are what are called the autosomes. You can see that we have 2 copies of each chromosome because we are diploid. We have pairs of chromosomes. So we have 22 pairs of autosomes, A-U-T-O-S-O-M-E-S, autosomes. And then we have a sex pair, right? And so that would make 23 pairs of chromosomes, which is why that company is called 23andMe, right? The one that does genetic testing. And so it's because we have 23 pairs of chromosomes. But this is extremely important. Pay attention and listen up. We have 23 pairs of chromosomes, but there are 24 types of chromosomes in the human genome. And so you can see that here. So we've got 1, 2, 3, all the way up to, so 22. Right, 22 pairs, and 22 types, therefore, because these are basically homologous chromosomes. They're not identical, but they're pretty much the same in terms of what genes they have. They just have slight variation among those genes, right? So among those alleles of genes. So we have 22 types, and then we have 23 and 24. There are 24 types of chromosomes. 23 pairs, but 24 types of chromosomes. Okay, so the sex pair is comprised of an X chromosome and a Y chromosome. The X chromosome is absolutely essential to human life. No human being can survive without an X chromosome. You can be perfectly healthy without a Y chromosome, right? You could ask any XX female. She is perfectly healthy without a Y chromosome. But of course, you could actually even survive without a second sex chromosome. So you could be XO, right? So you just have one X chromosome and then nothing over here. And so that would be Turner syndrome, XO. But you cannot survive with just a Y chromosome. You have to have an X chromosome. So that is a karyotype. You can learn a lot from a karyotype. Like, for example, this individual does not have Down syndrome. Down syndrome is also known as trisomy 21. So if there were 3 copies of chromosome 21, then we would see that this was a trisomic individual, but that's not the case. And, you know, to a trained eye, these chromosomes look, not that I have a trained eye, but to a trained eye, these chromosomes, you know, I would have guessed, look pretty healthy. And so you have a healthy human, apparently genetically, or I should say chromosomally male human being. We'll see a little bit later on in the course that just because you have a certain set of chromosomes doesn't mean you're going to be that sex, right? So there's a difference between, so we're not talking about gender, we're talking about sex, right? So there's chromosomal sex, there's gonadal sex, there's physiological sex, right, which we call the phenotype or phenotypic sex, right? internal versus external genitalia and some other features. But basically, this would be an old-fashioned karyotype. And so karyotypes are valuable, right? So basically, observations of chromosome behavior are what placed certain genes on certain chromosomes. And so cytogenetics is now currently used to map where genes are located because, again, we have 25,000 candidates and we've sequenced the human genome, so we think we know where everything is. But if you want to know how a gene interacts with another gene, right, that's when you would start to use something like cytogenetics. You could also study chromosome structure, and you can also identify abnormalities. We'll talk about all sorts of chromosomal abnormalities that can lead to all sorts of different outcomes a little bit later on in the course. Actually, there you go, in chapter 6. Now let's look at a modern karyotype, right? So this is, I worked with a woman who was just an absolute artist when it came to this. She made it look so easy. This is called FISH, fluorescence in situ hybridization. We have chromosome-specific fluors that can be conjugated to each one of these chromosomes. Basically, you could paint these chromosomes with a different fluorescent dye. And these dyes are very, very different to the filtered eye of technology. So like, for example, you probably can see the difference between this yellow, and this yellow. I would go with, I don't know, mustard yellow. I guess I'll go with canary yellow and banana yellow. I don't know. I'm just making stuff up. But the point is, these are 3 different yellows. They have different wavelengths of light. If you're not familiar with fluorescence, you hit it with one wavelength of light, and then a separate wavelength of light is emitted. And so you can use filters to just filter out and detect each chromosome based off of what wavelength of light they emit when they're excited. And so you can certainly easily tell each chromosome from one another. You can paint the chromosomes different colors, and you can learn a lot from a karyotype, because here is the karyotype of a normal cell taken from a woman, an XX female, that was being tested. And so she was being tested. She had a tumorous growth. And so they did a biopsy of that tumorous growth, and they wanted to know, basically, is that tumorous growth relatively healthy cells that are just growing at an alarming rate, which is certainly possible, or could it possibly be a cancerous tumor? Right? And so if you look at a karyotype done from the biopsy of her tumorous growth, I believe the scientific description of this would be it is completely jacked up, right? So what you're looking at is all sorts of wrong, right? So there shouldn't, so obviously this was a cancerous cell. So if you start, rapidly progressing through the cell cycle like a cancerous tumorous growth would do, then there wouldn't be proper time for homologous recombination or replicating your chromosomes properly, et cetera, et cetera. And so you end up with, so one thing you should never have is, like rainbow chromosomes, right? You shouldn't have chromosomes that have multiple colors. Here's a good example. Here's purple, then some blue, then some pink, and then this one has white, right? And then, so you should never have multiple colors like that. Like this is clearly not correct. This was chromosome 9, right? Chromosome 9 was mostly that color. Now it's, that's not even the right blue, right? that looks like chromosome 21 material. I don't know. So, so basically, that would be, evidence that that certainly is not a healthy cell. You can see the value of cytogenetics in terms of identifying the chromosomal abnormalities that may exist in certain cell types. And so then we get to molecular genetics. So you can see the value of cytogenetics in, identifying chromosomal abnormalities that exist in certain cell types. But then if you want to go even further, if you want to drill down even further, then you would get to the molecular level. And so molecular genetics are what we're going to study towards the tail end of the course. And so molecular genetics had, has had the greatest impact on human genetics since the early 1980s. I just told you about Herbert Boyer, Stanley Cohen, and Genentech and coming up with human insulin, right? So that's what I mean by the early 80s. That's when that was done. So molecular genetics concerns the chemical nature of the gene itself, right? How genetic information is encoded, how it's replicated, how it's expressed. So we're going to talk about replicated. Okay, so that's going to be, we're going to learn about DNA replication, right? How it's expressed is going to be transcription, translation. How it's encoded is going to be just deciphering the genetic code. I believe that's chapter 8. So there's just a number of different things we'll be talking about when we get to molecular genetics. But the basic concept of molecular genetics is that knowing the chemistry, knowing the biochemistry of these DNA molecules and DNA and as well as RNA and all the other molecules that are involved, the enzymes, allows us to do quite a bit of not just studying, but then actually repurposing this material. Because basically what we've done is we've discovered the code, right? And so now that we know how to program cells by writing their code, or we know how to read the code, we can rewrite the code and reprogram cells. So molecular genetics includes the cellular processing or processes of things I've already said, right? Replication, transcription, translation, and perhaps most importantly, gene expression. So we'll talk about regulation of gene expression. We'll talk about mutation. We'll talk about all sorts of different things towards the end of the course. So the focus for molecular genetics is going to be on the gene and its structure, its organization, and its function, the biochemistry of it. And then, of course, here's just a really rudimentary idea or sorry, graphic of how you can cut and paste, different DNA sources together. That's called recombinant DNA technology or genetic engineering, more colloquially. So molecular genetics utilizes recombinant DNA technology to identify, isolate, clone, and analyze genes. Why would you want to cut and paste? You can clone one particular gene, put it onto a little vector, stick it into E. coli cells, and you can study that one gene out of context, right? Instead of like trying to listen to one instrument in that sea, that orchestra of instruments, you could then just, basically shut everybody else off. You take that one instrument, you take that musician with their instrument, you basically remove them from the orchestra. You go and you get to hear them play solo. That's how you can actually identify what it is that gene does, and that you could get more information about that gene. You could figure out that gene. You can basically then theorize how it is going to interact with other genes, and then you can go back and check in context. So clone genes are very important because they give us insight into how genes are organized and how they work. And so they also can be transferred between organisms. And I mentioned this earlier in the lecture, right? So they can be transferred between organisms in order to massively produce a particular product, to enhance certain other, stuff that we buy, right? And then of course there's also the aspect of gene therapy, right? Theoretically, we could replace one gene with a non-functioning gene with either a human copy, a functional copy of that gene, or some other gene from some other location that can do the work, right? Sort of like using a particular heart valve from a pig's heart to replace a heart valve in a particular patient. And so it's sort of like that, except at the genetic level. Lastly, we have the last class that, we're not going to get to chapter 18. Our quarter is just too short. So I will just pepper in little instances about population genetics. I've already done so with cystic fibrosis and being of Asian descent or being of my wife's European descent. And so population genetics studies the inherited variation in population of individuals and the forces that alter gene frequency. So here you can see, like, this is just a random example of population genetics. It doesn't actually, it's based off of data, but it's an artist rendition. But there's the A allele, right? And so you can see the percentage frequency. You can see not very much in South Americans, whereas you could see that there is a tremendous amount in certain, that's interesting, right? You have nothing and then you have a lot. So that would be a very interesting finding. You'd be like, what's the difference, right? And so you might find that this is a very isolated population, or there is no population there, right? There's just no people there. And so then you can see it's very concentrated around here. Um, so you can see also down here. So you can look at genetic variation, genetic drift. You can look at population, migrations. It basically population genetics defines the extent of genetic variation within populations, and it also explains how migration, population size, natural selection, all sorts of catastrophic phenomenon, all sorts of things affect variation. What for those of you that are animal sciences majors, you may have heard of the Ngorongoro crater in Africa, where it is a very, when I say crater, don't think of like just a little crater that you can see from end to end. You can't see the the rim of the crater all the way around. It's huge, and so there are populations within it that have migrated in. So like lions and giraffe and gazelle, right? So impala, whatever it may be, and Cape buffalo and all that, and they're down in there, and they have been isolated genetically from the plains lions and zebras and whatnot. And so they are starting to drift and genetically drift, and there may not be completely different speciation yet, but they are certainly, you can see a difference if you look at the population genetics data, and you can then go, oh, okay, well that falls right in line with this crater, and so you can actually, like for example, this is certainly not the case, but what actually I don't know. Well, this is just A allele. This is not real. But if you saw something like that, right? So like 15 to 20, and then under 5, right? So then you could see how like this might be the Ngorongoro crater, right? Actually, I would guess this would probably be more likely the Ngorongoro crater. But the point is, you can see little pockets of a lack of a particular genetic, or a lack of particular allele in certain populations.